Hi everyone. I'm Krystina Hess. I'm a SMART student and a PhD candidate in Chris Jewell's lab at the University of Maryland. And in our lab, we focus on using biomaterials-based systems for disease treatment, specifically in autoimmune disease and in cancer. And my project specifically that I'm going to talk to you today about is using quantum dots, which are a nanotechnology, to deliver immune signals and to promote immunological tolerance rather than inflammation in the autoimmune disease multiple sclerosis. So just to start by familiarizing you guys with some of these immunological terms that I'll be talking about in this presentation. First, an antigen, something like a pathogen, or maybe something that's being delivered in a vaccine. It enters your body, and then it can drain to the lymph node passively, or a cell called an antigen-presenting cell can pick it up and bring it to the lymph node. There, it's going to present this antigen to T cells, and that T cell can either become a cytotoxic T cell, which will go out and kill infected cells, or it can become a helper T cell, which will help B cells produce antibodies. And sometimes your immune system malfunctions, and things like T cells and antibodies can actually recognize parts of your own body as foreign, and those become the antigens. And this will lead to autoimmune disease. I'm sure you're familiar with some of these examples like multiple sclerosis, rheumatoid arthritis, type 1 diabetes. And so some of these the treatments that are available for some of these diseases are not curative, meaning you might have to inject them every single day. And they're also typically broadly acting immunosuppressants, meaning that they're going to shut down your entire immune system rather than the specific response. And that can lead to immunosuppression, and this can leave you open to opportunistic infection. So what we're trying to do, us and many other researchers, is induce these regulatory T cells, a type of T cells that specifically suppresses a response to something like myelin, which is attacked in multiple sclerosis, rather than shutting down the entire immune system. So as I mentioned, these antigen-presenting cells and T cells interact in the lymph node, which you can see here. They're basically being programmed to respond to an antigen either in an inflammatory way or a regulatory way. So what we're hoping to do by developing therapeutic vaccines is to reprogram this response so that the T cells will stop attacking myelin and instead the immune system protects. Biomaterials offer a unique opportunity to deliver immune signals for these therapeutic vaccines. And some of these reasons include ability to deliver multiple signals at once, like a self-antigen and a regulatory signal, which will change the way the body responds to it. They can also offer cargo protection. They can change the way that the immune signals are trafficked through the body and what kind of cells take them up. And interestingly, some groups have recently found that in the absence of any other immune signal or any type of drug, delivering just the antigen that's being attacked in an autoimmune disease in some sort of particle form can actually lead to tolerance. So one last thing to motivate my project is that some recent revelations have come out that there is an actual connection between the lymphatic system and the central nervous system, which we didn't know about before. We weren't really sure how T cells got in and out of there, but these groups have found these functional lymphatic vessels that allow immune cells and fluids to travel directly from the central nervous system to deep cervical lymph nodes and back. So what we're wondering is, can we engineer a response to myelin in multiple sclerosis in the lymph nodes, create regulatory T cells, and then have those T cells migrate out of the lymph nodes to the CNS and control that pathogenic response. So the technology that I chose to use for this approach are quantum dots. They're ultra small particles, as you can see in this image, and they're specifically advantageous because they can be decorated with proteins and peptides, and you can put a large amount on them, and it's highly controllable how much you put on there. So we decorated quantum dots with the MOG peptide, which is myelin oligodendrocyte glycoprotein. It's part of the myelin that's attacked in a mouse model of multiple sclerosis. And we saw that even with up to 100 of these peptides on the quantum dots, they maintain this really small size. This is great because this allows them to quickly drain right to the lymph nodes where we're hoping to engineer that response. We also found that we could control the charge of them just by adding more or less of this positively charged peptide onto the quantum dot, as you can see in this migration through an agarose gel. So to see how functional these quantum dots were, we moved into primary immune cells and first gave them to dendritic cells, which are a type of the antigen-presenting cells I mentioned earlier. And we found that Some people are a little bit afraid of quantum dots as being toxic, but in this system, they had almost no effect on cell viability. And these antigen-presenting cells are supposed to take up the antigen and then present it. So we first tested, can they take it up, and used flow cytometry and found that just in 2 hours, see a really large signal from these very fluorescent quantum dots, and it's dose dependent. And then for the next step in the immune cascade, we added in transgenic T cells. And these T cells have receptors specific for myelin being presented by the dendritic cell. So in response to seeing this antigen and some sort of inflammatory cue or an adjuvant, they should proliferate. So we found that regardless of if the peptide was delivered alone or on a quantum dot, the cells proliferated. So this is a viable way to deliver the antigen and have immune cells still be able to recognize it. So again, taking advantage of the fluorescence, we also took out the lymph nodes of injected mice. We took out the inguinal lymph nodes on the injection side, which we called draining, and also on the non-draining. And in just 24 hours, you see an extremely bright signal in the draining lymph node and even a little bit in the non-draining lymph node. And this can be quantified by flow cytometry, as you see a very high signal there. Next, moving into our actual disease model. The mouse model of multiple sclerosis is experimental autoimmune encephalomyelitis. And it's a model of progressive multiple sclerosis, which is typically seen in the later stages of MS, as opposed to relapsing and remitting. This neurodegeneration and paralysis associated with the neurodegeneration continues to get worse. And you can score it from 0 to 5 with how severe the paralysis is. So an untreated mouse typically has a score of about a 3 at the peak of disease, which is around day 16, meaning that their tail and their hind limbs are completely paralyzed. They have to drag around the rest of their body just using the front limbs. But we used our treatment mechanism, the MOG-coded quantum dots. You can see the disease severity goes way down, and that's these red dots over here. And it also affected the incidence. At peak of disease, which I mentioned around day 16, only half the mice even showed any symptoms at all. And this is one of those mice that showed no symptoms following this treatment regimen. It was just able to move around freely without any impairment to the tail or the hind limbs. Next, wanted to understand the role of the dose of the MOG. As I mentioned, you can put a lot on the quantum dots and you can also control it. So we treated with varying MOG dose on the same number of quantum dots. And we found that there was an impact in how much it can control the disease. You can see as you added more MOG, which is the red dots, there was a better control. And this is extremely evident in the incidence, where at the end of the disease course, only one mouse had any symptoms at all. And next we wanted to see, is it just the dose or is it also impacted by the ligand density? So this time, instead of varying the MOG, we varied the amount of quantum dots. And we saw there, if you put 25 ligands per quantum dot, which was spreading out the amount of MOG on more quantum dots, versus 65 MOG ligands on a quantum dot, which is just a higher density, we saw there was a large difference in how that disease was controlled. So we found that more MOG spread out on more particles maybe was reaching more places and then was more effective. And this was also extremely evident in the peak disease, which we looked at, and in the incidence. We see the same trend. So moving forward, we have a couple of questions, mainly to address what's the actual mechanism of this tolerance. So we found that it does work in controlling disease, but we want to know why. So one thing we can do is take out different organs, lymphoid organs such as the lymph node, and look for where the quantum dots are going within that organ themselves. So in this pilot study, we found the quantum dots in red were co-localized with Marco-positive macrophages. And these are thought to be involved in tolerance because they're typically involved in clearing apoptotic cell debris, which could become a self-antigen. And another way we can do this is look at the types of cells that are recruited to a lymph node once the quantum dots get there. In one of the studies I showed you earlier, we took out the lymph nodes right around the onset of disease and found that a high dose of the MOG quantum dots induced a large amount of regulatory T cells into that lymph node, which are, again, thought to be one of the control mechanisms for this disease. And with that, I'd like to thank my lab. I'd like to thank our funding sources, especially the DoD SMART Fellowship, which is funding my dissertation.